Now, hello Team IBBC 36. Welcome to the Global Youth Science and Technology Board 2021. I'm Kimton, the MC of this judging panel. This team is from India, a novel study of the biopesticidal properties of Anola reticulata. First, we would like to thank you for attending the judging online today and for your efforts made in the past few months for developing your project. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or any other materials. We will remind you the time after 5 minutes, and you have to stop your presentation when time is up. And there will be 4 minutes of Q&A session after your presentation. So do you have any questions?
No, sir. Thank you so much.
Hey, if no, please share your PowerPoint now. Hey, you can
Good afternoon, everyone. Good afternoon, everyone. My name is Sarvesh Prabhu, and the name of my project is a novel study of the biopesticidal properties of Anola reticulata, more commonly known as Ramphal. The main aim of this project is to find a viable alternative to chemical pesticides and insecticides, and we also want to study the biopesticidal properties of Anola reticulata. We already know what pesticides have done to the environment. They are also a leading cause of air and water pollution. Unknowingly, the farmers have sown the seeds of the demise of their own health and land. Therefore, we need an efficient and cost-effective alternative to pesticides and insecticides. The preparation process of the extract is extremely simple. We pick out the fresh leaves, cut it and grind it into a paste. We filter it, dilute it, and then apply it on various plants over the interval of 4 weeks. In my hand, you can see the extracts. The first plant that we tried it on was bittergourd or Mormodica charantia. We had the issue of stunted growth and damaged fruits due to the burrowing of insects. This problem persisted in the control plant. Here you can see the trial plant, which had healthy fruits, which were long and sharp. The second plant that we tried it on was curry leaves or Murraya koinigi. We had the issue of caterpillars and whiteflies eating away at the leaves of the plant. Here you can see in the control that the caterpillars and whiteflies persisted, while the trial plant was free from both pests. Here you can see that over the period of 7 days, there is eradication of the population of whiteflies on the curry leaf plant. We also tried it on several other plants, including tomato, hibiscus, and rose. We noticed that the harvest was better in quality and larger in number for the simple reason that the leaves were not being eaten by various pests. Here is the chart of days needed for the eradication of pests for various plants. Hibiscus takes the least number of days, approximately 3, while tomato takes the most number of days, approximately 12 days. With pesticides, there is always a concern of toxicity as the products on which it is applied are ingested by us humans. The mammalian toxicity report conducted by the University of Mysore concluded that it does not have any significant toxic effect on mice. However, it is extremely potent against brine shrimp. Currently, we are also underway a brine shrimp lethality assay as we speak. The first test that we did was a degradation test in which the extract was kept in various physical conditions. We concluded that the extract must be kept in the shade and can be applied to the plant twice, as it is a 2-week interval before it is unusable. The second test we conducted was a very basic pH test. We noted that with time, the extracts moved towards the acidic side, and the extract of the seed was significantly more acidic than the extract of the leaf. The third test that we conducted was the component test. We realized that the extract is rich in minerals such as zinc and magnesium, and it also contains ammonium and oxalate ions in high concentration. The fourth and last test that we did was on the antimicrobial test, which was on E. coli bacteria. We realized that Anola reticulata shows good antibacterial properties comparable to that of garlic, another organic pesticide. We believe it works due to acetogenins, a biochemical which are present in the Anonaceae family and are mitochondrial poisons, inhibiting cellular energy production. They also act as antipedant, and therefore the insects die after eating the plant. The four main acetogenins present in the plant are 14-hydroxy-20-deoxyrhodinosin, squamosine, neoanine, as well as anonasin. In the Indian context, it drastically reduces the expenditure by farmers by approximately 80%, and the fruit of the plant can also be sold for additional income. Here is the cost per acre of the crop of cotton. If we were to use traditional pesticides, the cost would be approximately 1400 rupees or 150 Hong Kong dollars. However, if this biopesticide is used, the cost reduces to simply 120 rupees. And with the additional income of fruit, the total benefit for the farmers is actually 261 Hong Kong dollar per acre. Its application is in four main regions: whiteflies, mosquito larvae, caterpillars, as well as aphids. There are several biopesticides in the market. However, this has an advantage over it, as it has a much wider applicability. It has many volatile compounds, thus the residue does not linger for long. It is easy to prepare and cost effective, as it costs simply 2.65 Hong Kong dollars per liter. We also conducted a survey of several farmers and a few house gardeners. We realized that they do not know the name of the pesticides they are using and use it without proper protection. In the future, we want to test its antioxidant qualities, isolate these compounds and test its efficacy, and collaborate with farmers in our region.
Thumbs up, student. Okay, we will have the Q&A session now. Judges, do you have any questions?
So maybe I can start my question first. So does anyone suggest that the Anona reticulata can be used as biopesticides before? So is it your original idea, or that has been done by some other else?
So this is my original idea. I actually skipped that slide, which was on how I got the idea. It was basically, I was sitting on the terrace and we have a small house garden. And I realized that there were many pests, but only one plant was free from all kinds of insecticidal pests, not microorganisms. And I thought, what makes this plant special? And I made a crude aqueous extract and used it. And I noticed significant results, after which I went to test it and made ethanol extracts of the same.
So how do you determine the dilutions of the extract you use in your plant?
So we are using dilution 9:1. That is 9 part distilled water, 1 part concentrated extract.
Here so how do you know it is too concentrated or too diluted? How do you test this? It's a right concentration.
So this is a standard protocol that we use for all pesticides. So it is always 9 part of mixture and 1 part of active ingredient. So we follow the protocol for the same.
Okay.
However, we also tried it with various other dilutions like 50%, but we realized that it leaves stains on the leaf if we were to use different dilutions.
Professor Lam, do you have any questions? You have muted yourself. Please unmute your microphone.
So very nice project. Congratulations. And I have 1 or 2 questions for you. And
yes,
now from the photos that you have shown us, can you comment on whether your new biopesticide is a systemic or local pesticide? You know what I mean? For systemic pesticides, you apply your pesticides on one part of the plant, then the whole plant got protected. But now, of course, for local pesticides, you apply on maybe on the fruits, then the fruit's protected, but not the leaf. If you apply on the leaf, then the leaf's protected, but not the fruit. So can you comment on what's the nature of your new biopesticide?
So we usually have to apply it on every part of the plant, except the stem, of course, the leaf as well as the fruit.
Okay, so have you ever tried like just applying on the leaf to see whether like other part of the plant still being affected by those insects or pests?
Yes, sir. On 2 plants, hibiscus and rose, we did not apply it on the flowers per se, but we applied it on the leaves. We noticed that the leaves were protected, by the stamen and the pistil of the plant was constantly being eaten because it had...
yes, it's more like a local pesticide.
Yes, sir.
Okay,
yes,
very
sir.
good. Now, I've also seen that you have done some toxicity tests using like microorganism, using E. coli.
Yes, sir. The antimicrobial test.
Yes. But I think, I mean, your new biopesticide is more like an insecticide because you have already proved to us that, I mean, it's quite effective on some like caterpillar, like white flies. These are actually rather large insects.
Yes,
so,
sir.
so the toxicity, the nature, the mechanism may not be the same. And
Yes.
so have you ever looked into the exact mechanism of how your new biopesticide kills those
the insects or the microorganisms, sir?
Insects?
Yes, sir. I just need to go to this slide. Yes. So here they basically inhibit energy production in the cells. So ATP needs to break twice and they do not allow that to happen. So the protein needed, NADH ubiquinone reductase, is actually not made because of these compounds. They act as a substitute in the mitochondria of the cell.
Okay, thank you, team. Okay, time's up, I
Thank you
think
so
now.
much.
Okay, you may leave the meeting room now and see you tomorrow at the award ceremony. Bye-bye.
Nice work. Thank you.
Thank you.
Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. This team is from Philippines. The project is Paper 1, the utilization of biowaste extracts on non-wood material for water-resistant paper production. First, we would like to thank you for attending the judging online today and for your efforts made in the past few months for developing your project. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or any other materials. We will remind you after 5 minutes and you have to stop the presentation when time is up. And there will be 4 minutes for Q&A session after your presentation. Do you have any question?
Answer.
No, you can share your screen now.
All right, I'll just share our video.
Okay, you can start when it is ready.
Is it already seen?
Not yet.
It's a black video.
Yes.
Okay.
Black video.
We will start in 3, 2, 1. Paper, a commodity used worldwide, often utilized for writing and packaging. The production of paper, however, depends on the deforestation of trees around the globe, which causes drastic consequences such as flash floods, habitat loss, and climate change.
Specialty paper, packaging, and even the currency notes are only some of the papers produced by the paper industry. However, some of the manufactured papers use chemicals that are considered harmful to the health of people. This can cause diseases such as cancer and eczema.
Why use polymer?
Product, different testings were conducted on our paper.
For the water absorption test, results show that uncoated samples absorb more water compared to coated ones. For thickness, data suggests that coated samples are always thicker due to the biowax on the surface. For grammage, this is the weight of the samples. We found that coated samples weighed heavier due to the added layer of biowax. The delta's average reveals that the average of coated samples is higher than that of uncoated ones. Burst strength was tested on both sides of the paper. It indicated that the coated samples had the higher average. Our own porosity test proved that the taro biowax coated abaca paper can still absorb ink, regardless of water resistance. We therefore conclude that there is a significant difference between the two papers, with the taro biowax coated abaca paper having a better water resistance property than that of regular abaca paper.
Our funding agency for our project paper 1 initiated a project which aims to improve the Philippine peso bill using biomass materials such as abaca.
The proponents of the proposed project for the Philippine peso bills are the qualified scientists who have assisted us all throughout the process of doing the paper 1.
These peso bills in our hands will soon undergo innovative changes, more eco-friendly, burst-resistant, higher tearing strength, and more water-resistant. Gone will be the days of chemical-infused paper currency, and we, the proponents of Paper 1, will be one of the proponents for this innovation. Currently, our paper was accepted for peer review and will soon be published.
We were given a chance to make a difference, and we stood up to the challenge.
Research is indeed difficult, but all of these sacrifices have paid off.
This is one simple innovation for the paper industry, and we, the Paper 1 team, take pride in being part of our country's symbol of prosperity and security.
With pride and honor,
this is our humble project.
Paper 1, the utilization of biowax extracts on non-wood material for water-resistant paper production.
Okay, now we come to the Q&A session. Any questions from our judges?
Okay, maybe me first. And so very interesting project. Thank you very much. And I learned a lot of things from reading your report. Now, one thing I would like to learn more is that, is in ordinary paper production, papers usually have this wax layer. Is this a must for like office paper?
Actually, it's not just for office paper. We have things such as parchment paper, which have a layer of wax on top of them called paraffin wax. According to our study by Yetman 2020, it is actually revealed that when you burn the wax on paraffin, it is actually going to release chemicals such as benzene and petroleum, which is harmful to whoever uses it. So we need to fix that kind of toxicity. And aside from water resistance, our paper is also very good in terms of burst strength and tearing strength. So it's not just good for this type of paper, but also for all types of paper.
Okay, very good. Now, this leads to my next question. Okay, now you extract your wax from taro using cyclohexane.
Yes.
So I think the bio wax that you obtain is actually quite hydrophobic, right? They
yes,
they hate water, more like non-polar. Now I don't know whether you have looked into whether your bio wax really able to wet your paper. Okay, and I also don't know the nature of the paper, whether the paper is kind of have more polar group on the surface, it's like more hydrophilic or hydrophobic. And how are you able to ensure that the wax that you put onto the paper adheres to the paper without like falling off?
Yes, well, first, according to NASRI 2014, it is actually proven that octacosanol is the main reason why taro has this hydrophobic property. It is a nonpolar solvent, so we use it is a nonpolar fatty acid, so we use a nonpolar solvent such as cyclohexane, which was guided by our chemical consultants, Ma'am Mildred Fidel, Ma'am Melvi Emprese, and Ma'am Dr. Gladys Completo. This is actually the reason why we had the water resistance test. However, we wanted to opt for the hydrophobic test, which is ISO 4096. Which actually tests for the contact angle of our water droplets. However, we were unable to attain this at the time, and we opted for the water resistance test instead.
Okay. My question is, how are you able to ensure that your wax can adhere onto your paper? Is it a problem, or is it not a problem at all, that the wax really can stick to the paper?
When we actually tested our results, we ensured that the paper was dried for at least a few hours in order to avoid it from getting slipped off. That's why when we had our testings, as you could see in the photos and the videos, it is actually revealed that the water resistance property and the other tests are still effective, even though it's already dried because it's sticked.
Okay, so it won't peel off.
Yes, it won't peel off.
Thank you.
We have 20 seconds left, so maybe I'll give you some comment or suggestions that I think it may be important. So in addition to compare with uncold paper, so it will be interesting to compare with some commercial available water-resistant paper so that you can know the quality of your paper when compared with those that in the market. And so you may know how to improve your products to make it more marketable acceptable, right? So I think it's important to have control in your experiment. Yes, so I'm happy to see you have some statistical evaluation in your data. So it's good. It's a good thing.
Actually,
yes,
for
sir,
our
we will take note of that.
yes.
Hey,
actually,
thank you, teams. Hello.
Yeah.
Okay, time's up, I think. So you may leave the meeting room now and see you again tomorrow at the award ceremony. Bye-bye.
Thank you so much. Have a good day and stay safe.
Have a good day.
Hello, Team IBBC 40. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kim-tan, the MC of this judging panel. This team is from Indonesia. Innovation of green biodiesel by trans-esterification reaction of black soldier fly larva fat with calcium oxide catalyst from Pila and Pulasia shells, a very long name. First, we would like to thank you for attending the judging honor today and for your efforts made in the past few months for developing the project. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or other materials. We will remind you after 5 minutes and you have to stop the presentation when the time is up. And there will be 4 minutes for the Q&A session after the presentation. Do you have any questions?
No.
If no, you can share your PowerPoint now.
Okay. Wait. Hold on.
Okay. Okay, we can see now. So you can start your presentation when you are ready.
Is the presentation already shown in the screen?
Yes.
Okay.
Can start anytime.
Okay. Good afternoon, judges and audience. We are from SMK Kanusa Bangsa, Bogor, Indonesia. I'm Raffi Irsyad Saputra and my partner.
I am Siti Sulistiani.
In this moment, we're going to explain about our project with the title Innovation of Green Biodiesel by Trans-esterification Reaction of Black Butterfly Hermetiolucens larva fat with calcium oxide catalyst from Pila ambulacea shells. Okay. When we hear about larva, it must be something disgusting. But black butterfly larva contains high lipids that can be made into biodiesel. Commonly, biodiesel production process by trans-esterification is react lipids and methanol using catalyst. But methanol has high toxicity.
The catalyst is usually sodium hydroxide or sulfuric acid. But sulfuric acid can increase acid number and make corrosion to the machine. Sodium hydroxide can form salt, so hard to separate.
So, in this innovation, we use ethanol with lower toxicity and calcium oxide catalyst from Pila ambulacea shells. Pila ambulacea shells less utilized, but it contains high calcium carbonate, calcinated to form calcium oxide, which is less soluble and can be reused. So, don't give me a problem in pollution.
These are the tools and materials that we use.
This is our research design. Pila ambulacea shell floor calcinated and characterized by Fourier transform infrared. Meanwhile, black butterfly floor extract the fat, continue with trans-esterification, then characterized using GCMS. The reaction was done by 3 treatments: mole ratio, time variation, and catalyst weight. The product tested by acid and saponification number.
The biodiesel production process done by mix calcium oxide catalyst with ethanol. After that, we accept larval fat added drop by drops and heat on 60 degrees Celsius. And then separate and wash the biodiesel. Then dry by sodium, sulfate, and hydrate and filter it. Biodiesel product analyze the acid and saponification number. The result of Fourier transform infrared analysis shows that calcinated Pila ambulacea cells doesn't obtain CH bond, but it has OH bond.
And based on the characterization of black butterfly larva fat with GCMS, it gained that the black butterfly larva fat contained fatty acid with 10 until 16 carbon atoms.
The data of acid, saponification, and ester number was tested by ANOVA and give the result that the mole ratio and duration time variation didn't give significant difference. cause the p-value is more than 0.05. But the catalyst weight variation gives significant difference, with the p-value is less than 0.05, which on the 0.75 gram catalyst gives the lowest acid number.
This is our biodiesel waste management. So, from this research shows that the black butterfly larvae contain fatty acids with 10 up to 16 carbon atoms, and based on the quality analysis, shows that the mole ratio 1 to 6 and 1 to 8 didn't give significant difference. Duration time 2, 4, and 6 hours didn't give significant difference, but the weight of catalyst gives different quality to the biodiesel, and the biodiesel production waste safe for environment.
These are the reference that we use. Thank you for the attention.
Renewable energy for the better future.
Thank you. Okay, we will have the Q&A session now. Any questions from our judges?
Maybe I can start with my question first. So it's an interesting project. So thank you for your presentation. So I just wondering, why do you come up with this idea to use the fly and shell to make biodiesels? Are these material easy to collect? Can you like put them into mass production? So how about the cost effectiveness of using this material to make biodiesel?
The cost on this.
Several questions. First of all, is how you come up this idea. The second question is that is this material easy to get? And third is the cost effectiveness.
Okay. I will answer the first question. Why do we choose this innovation? Is because in Indonesia, the black butterfly larva hasn't utilized optimally. And we found that in China and Saudi Arabia, they already developing the black butterfly larva to become a biodiesel. And because it contains high lipids, so we interested on their research, and we developed from the previous research who conduct by Liang Zheng and friends from China and Hong in Saudi Arabia. The reason why do we choose this research is supported by the fact that this black butterfly larva is very easy to get because we can find, because it already spread around the world. What I know from the literature review, the black butterfly is already spread in Australia, Russia, China, South America, and in several countries in Europe. So that's the reason why do we choose the black butterfly. I want to add my answer is it's supported by the life cycle of the black butterfly is short. So we figure it out the optimal utilize with this larva. And for the raw material is very easy to find. Once I said that the black butterfly is easy to find because it's already spread in other country, and it can live in in many climate substance and in many circumstances. And for the catalyst is we use the pila ampullaceae shells, which in our country, especially, and in Southeast Asia country, it's very easy to find because the pila ampullaceae shells is the shells we can get it from the rice field. Yeah, but this innovation we use the calcium oxide. We can replace the pila ampullaceae shells with another materials who contain more calcium. And for the cost of this research is we don't pay for many costs because we found out that the pila ampullaceae shells is in our country is a waste because it consumed by the citizen in Indonesia. So we take it to make the biodiesel. So that was why the cost on this research is cheap.
Mm-hmm.
Yeah.
Okay, thank you, team. You may now leave the meeting room and see you tomorrow at the award ceremony. Bye-bye.
Thank you so much.
IBBC 01, welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. This team is from Ukraine. Study of efficiency of corrosion inhibitors properties created on the basis of plant raw materials in different environments. First, we would like to thank you for attending the judging online today and for your efforts made in the past few months for developing the project. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or any other materials. We will remind you the time after 5 minutes, and you have to stop the presentation when the time is up. And there will be a 4-minute Q&A session after your presentation. Do you have any questions?
No, I haven't.
Okay, so you can share your PowerPoint or your screen now.
Okay. Yes, it's my PowerPoint.
Okay, if you are ready, you can start anytime.
All right. Good afternoon, dear ladies and gentlemen. My name is Yuchinska Anastasia, and I'm a Junior Academy of Science of Ukraine. It's nice to meet you. And today I want to present my research work, which names Study of Efficiency of Corrosion Inhibitors Properties Created on the Basis of Raw Materials Plants in Different Environments. The literature which was used in my research work and in this presentation, you can see on this slide. The next, as a result of corrosion destruction, about 10% of annually metal production is lost. So it's considerable to pay attention and create a household inhibitor that can help us to protect our environment and protect steel. Recently, the so-called green inhibitors have around in different plants. And our reached goal was to create a household inhibitor on the basis of rough materials plants, such as common yarrow, celandine, and Altea, and test it in different environments. You can see our plants in this slide. The next, also our project meets the global goals of the UN, and our research hypothesis it was that in this household we can create household inhibitor on the basis of water extract of tomato edible, and the first, to begin with, it was necessary to disable the process of corrosion and understand it. You can see on this slide the process of corrosion. So we use the method of obtaining Pickling's fluids. It's Ukrainian scientist Olhin's method. Plant rough materials, leaves and stems of plants were chopped into pieces and poured with a weak chloride acid solution of not more than 5% strength. Left for 7 days for extraction. 8 samples were prepared. Option A from fresh plants and option B from dry raw materials. When the extract was ready, there was prepared a pickling solution for study of its efficiency. The pickling solution was applied to low alloy steel. Here you can see our formulas for our research and our calculations. The inhibitor solution density was taken equal to the solvent. All test samples were 1 millimeter in thickness. Corrosion losses were evaluated by the gravimetric method on the sample weight loss during corrosion. So here you can see in more detailed information about the formulas which was adopted. All my working process is shown on this slide. So, all calculation by formulas you can see in these tables. The plates under study were immersed into 2 types of environment, marine water and acidic. In total, all data from the tables are entered into general graphs. To sum up, the greatest value of the coating protective effects is displayed by the extract of dried celandine and top of fresh tomato edible, slightly less displayed by dry Altea. These results are confirmed by microscopic examination of the surface of the experimental plates. In acidic environment, when using water extracts of the plants, the degree of protection is 90%. In sea salt solution, the degree of protection was from 91 to 93%. Based on the results of the calculation, per liter is 13% of solution. One can prepare 3 liters of pickling solution, 3,000 liters of pickling solution, which can protect 31 square meters of steel surface and will cost from now $0.25. Therefore, this type of inhibitor is cost-effective and their preparation doesn't require special user training. The technique is easy to perform. And also, I'm sincerely grateful to my best supervisor, Bondarenko Natalia, who once saw the chemical potential in me and started working with such a different person like me. And to my second supervisor, Selivanova Tatiana, who continue and breathe new life into our work. Thanks for your attention.
Okay, thank you. Now we are going to our Q&A session. Judges, do you have any questions?
So maybe I start first. Thank you very much for a very interesting question and interesting ideas. I just would like to ask, because I went through your report, I'm still not too sure about how did you apply your solution onto the metal surface, and how able are your solutions to adhere onto the surface to produce long-term protection?
Our inhibitor was applied to our steel plate with a little pen. It's yes, and
your solution wet the surface?
What?
And your solution is able to wet the surface. The solution is able to spread across the surface of the plate. Okay. And then, I mean, any drip off or anything? How thick is the film formed on the surface? Have you measured that?
1 millimeter.
One millimeter is quite thick.
Yes, yes.
Wow, okay. And may I ask about the mechanism and how do these kind of plant extract able to protect your steel plate?
Such as ferments, as alkaloids and polysaccharides protect our plates. These ferments has insulindine, altea, tomato edible, and it protects our plates from corrosion.
And so they are antioxidant, or they are reductants, or whatever. Why do they stop metals getting oxidized?
It's something like coverage on our plates, and it can help not to react with the environment, our plates.
Okay. Yeah. Have you any kind of measurement that you can prove that your plant extracts able to really do that? Can I use other things like other extracts or like mineral oil to cover the metal plates? Can I obtain similar effect?
Was chosen as the extract of plants that have the highest ferments of alkaloids and polysaccharides. So we choose especially these REV materials.
Okay. Thank you.
Okay. So may I ask some questions? It's also a very interesting idea. So how do you decide what kind of plant extract to be tested in your experiment?
Oh, once I was at my grandmother's countryside house, and I saw a lot of corrosion in it, and it was hard for that old woman to protect her metals. So I decided it will be a great idea for my research work, because I fall in love with chemistry, biology, and I think it's interesting.
Yeah, but there are so different kinds of plants. So how to decide which one is the best candidate to be tested?
I know that our scientist Olhin has that method, but in internet environment, in internet media, there wasn't such projects like mine that have 100% told us that especially these REV plants have this extract like alkaloids and polysaccharides. So I tried to explain it, and I did it.
All right, thank you.
Okay, thank you, student. So you may leave the meeting room now, and see you again tomorrow at the award ceremony. Bye-bye. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. This team is from Ukraine. Dependence of intestinal microbiota of white rats on the effects of glyphosate, saccharin, and sodium benzoate. First, we would like to thank you for attending the judging honor today and your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of any PowerPoint or other materials. We will remind you at the time after 5 minutes, and you have to stop your presentation when the time is up. And then there will be a 4-minute Q&A session after your presentation. Do you have any questions? Okay, if no, you can share your PowerPoint now.
Shall I demonstrate my presentation?
Okay, you do not have PowerPoint? Okay, okay, we can see it now. So you can start anytime if you are ready.
Yeah, I'm ready. Let me introduce my research work on the topic dependence of the intestinal microbiota of white rats on the effects of glyphosate, saccharin, and sodium benzoate. Today, a large number of substances are used, the effect of which on the microorganism is not fully studied, especially in the complex. Glyphosate-based herbicides are the most commonly used means of controlling rural vegetation in the world. The use of pesticides in the cultivation of crops creates a risk of exposure to consumers of harmful chemicals due to their residual amounts that accumulate in the body of the animal or in the crop during their cultivation. Therefore, it remains relevant to study the effects of glyphosate on animals and humans both alone and in combination with commonly used preservatives and sweeteners. The most common preservative is sodium benzoate. The antimicrobial action of benzoic acid and its benzoate salts is based on the ability to inhibit the activity of enzymes. Among synthetic sweeteners, the most well-known are saccharin, the sweetness of which is 300 times higher than the sweetness of sucrose. After analyzing the already known studies of the effects of each substance separately, which are conducted in European countries and in the United States due to the lack of evidence, we realized that these substances, in combination with each other, can also change the organs and systems of the macroorganism. Therefore, the novelty of our scientific work is the study of the effect of glyphosate in combination with saccharin and sodium benzoate on the intestinal microbiota. The aim of the study was to determine the dependence of the intestinal microbiota of white rats on the effects of glyphosate, saccharin, and sodium benzoate under experimental conditions. The experimental part of the work was carried out on the basis of the Dnipro State Agrarian and Economic University. The material for the study was the feces of the rectum of white rats, control, and 3 experimental groups, 5 animals each, which for 42 days were administered the drugs under study. The first group of animals, control, consumed clean water. The second group, rats, were given a 1% aqueous solution of glyphosate. Animals of the third group were administered 1% glyphosate solution with 1% sodium benzoate solution, and the fourth group with 1% saccharin solution. For each sample, 1 gram of the contents was placed in a sterilized box and 9 liters of sterilized saline was added. After preparation of all dilutions, 0.1 milliliter of solution was taken from each tube with a sterile pipette and placed in a petri dish with a suitable electoral medium, increasing each dilution 10 times more. Then rested the cups on tubes on the table for 15 minutes and then placed in a thermostat at 37 degrees Celsius. Sterile beveled agar media of Volkanovsky and Siemens were used to identify certain species of enterobacteria. The tubes were placed in a thermostat at a temperature of 37 degrees Celsius for 24 hours and then recorded the presence of certain biochemical properties of microorganisms. Culling counts were performed on all plates with media. The grown cultures were smeared after fixing the preparation on slides. Gram staining and microscopy were performed using the light microscope series by 1C1. The studies identified the main representatives of the intestinal microbiome of experimental animals. Bacteria of the genera Bifidobacterium, Lactobacillus, Lactococcus, Escherichia, Proteus, Enterococcus, Citrobacter, Klebsiella, Enterobacter, Clostridium, Staphylococcus, Candida. The first diagram shows the quantitative and qualitative composition of the intestinal microbiota found in experimental animals of the first group, control group, which consumed pure water. We took this data into account when conducting the experiment comparison. The most numerous group of intestinal microbiota was bifidobacteria, almost 90%. The number of Enterococcus was almost 7.5%, and Lactobacillus, the total number of Escherichia coli Streptococcus, was within 1%. Isolated colonies of fungi of the genus Candida, saprophytic Staphylococcus, Staphylococcus aureus, lactose-negative opportunistic Enterobacteria, Clostridium, the number of which was within normal limits. For the second study group, the diagram shows that the use of 1% aqueous glyphosate solution led to the spread of opportunistic Enterobacteria and fungi of the genus Candida, up to 1%. The number of other microorganisms remained virtually unchanged. The quantitative and qualitative composition of the intestinal microbiota of animals with the third group is also shown in the diagram. The use of 1% aqueous solution of glyphosate with 1% solution of sodium benzoate led to an increase in the total number of opportunistic pathogens of the family Enterobacteriaceae, as well as fluctuation in the number of bacteria of the genus Enterococcus species, almost 20%. Fungi of the genus Candida, up to 1%, the number of bacteria of the genus Pseudomonas, which exceeded the reference range of physiological values. And the corresponding diagram for the fourth group allowed us to conclude that the use of 1% solution of glyphosate with 1% solution of saccharin led to exceeding the reference range of physiological values of three genera of opportunistic Enterobacteria, as well as fungi of the genus Candida and bacteria of the genus Pseudomonas. The number of all other microorganisms hasn't changed significantly. According to the results of the study and taking into account the results obtained, food producers are recommended to minimize the use of preservatives and artificial sweeteners to provide complete and truthful information about the composition of products on the packaging. The obtained results can be used in conducting research of a similar nature, carry scientific achievements in understanding the issues of diseases of both infectious and non-communicable etiology in human and veterinary medicine. Thanks for your attention.
Okay, so it's Q&A session now. Judges, any questions?
So thank you for your presentation. So it is a very interesting study. So I may have some questions for you. So so it is about the concentrations of the glyphosate, saccharin, and sodium benzoate. So is it a real situation that we consume that high concentrations of this substance in the daily life?
Yeah, we took a high concentration at 1% for maybe 100 milliliters because we wanted to see the whole picture of intestinal microbiota of microorganisms that appeared in intestinal microbiota of people and animal, because the substances are used in products. Yeah, concentration is a bit maybe less that we took, but for this study, maybe we took these concentrations.
Mm-hmm. So you mentioned in your report there are 5 rats in the group, right? So but however, when you're showing us the data, it is it should be an average value, right? Do you see a variation between different animals in the same group?
Oh, please repeat the question. I don't hear well.
Variation. So like 5 animals in the group, are they exactly the same, or you find different results among the same group? The variations.
Yeah, we do. As I say earlier, we took 20 animals variations, 5 animals in each group.
Mm-hmm.
They don't have like special abilities. I don't know.
It's all right. It's okay.
Any more questions? How about Professor Lam?
Maybe I ask something not related to your project. May I ask, have you thought about or actually look into whether the profile of the microbials or the gut microbials in your right rat will change back to normal after the animals not eating the pesticides or food additive that you have given them?
Mm-hmm.
I know this research work was conducted collectively, and after these animals, we use these substances on animals. Then we just leave these rats, and they I don't know, they got health.
Okay, because I mean, people are now talking about the importance of gut microbiome. Okay, that actually affects a lot, like metabolism, even like for in human, in little children, maybe their mental development. Okay, but it will be interesting to see if these like pesticides or food additive actually have long-lasting effect on the gut microbiomes, or the effects only occur like a kind of short period of time during consumption, and the gut microbial community will go back to normal. I don't believe that. Okay, but it will be nice.
Thank you for your question. It's a good idea for future developments, future researches. I will work on this. Thank you.
Okay, very good, very good.
Okay, thank you, student. So you may leave the meeting room now, and we will see you tomorrow at the award ceremony. Bye-bye.
Okay, thank you. Goodbye.
Hello, Team IPBC 27. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. This team is from Russia. The project name is Panovit, Further Protein of Microbial Synthesis. First, we would like to thank you for attending the judging honor today and for your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing. We will remind you the time after 5 minutes, and you have to stop the presentation when time is up. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No, I don't have.
Okay, so you can now share your PowerPoint.
Can you see it?
Yes, you can start anytime if you are ready.
Yes, okay, and hello there, members of the OC committee. Hello there, judges. My name is Vladislav Urganov, and now I would like to tell you about my project, Panovit for the protein of microbial synthesis, which we produce in cooperation with Biopractica Ltd. And I would like to say that now one of the most greatest problem of mankind is overpopulation. And every year in our planet, there are more and more people which require food. And most of these foods are animal produces. And in this way, these animals also require food. And now we have two main technologies of producing food for animals. It's food from fish and from different other. And this technology is not eco-friendly, and we would like to create technology for production of feed additive based on protein or for bacteria growing on methane and probiotic microorganisms with stable quality and high profitability. And this is our main. And our product consists of two parts: it's protein, which based on methanotropic biomass, and probiotics, which we are going to grow up directly on the protein. And these two parts make our product better than the other, because firstly, it is eco-friendly production. Secondly, this product have high profitability, and it's independent from natural factors. It's a complex product because we have a protein and probiotics in one product, and also it's a balanced composition. And now I would like to tell about some results which we received in this step of working. Firstly, about the economy part, we made the project documentation and charter was drawn up, because it's impossible to exist the project without this documentation. It's need for investor, and it needs to the plant in comparison with we work. Secondly, financial model of production and the financial model of production was made, and some investment figures was found, because it's also important to prove the profitability of our project. Now about the biology part. Firstly, we made the isolated microorganism showed the ability to grow on the methanotropic biomass, and we knew that our cultures can grow up on the methanotropic biomass, and we can use it in the intended process cycle. Secondly, we tested our cultures amylotic, probiotic, and lipolytic activities, and it's need to us to know that our cultures is probiotic and to prove that their probiotics. Their probiotic skills. And lastly, we knew about antibiotic resistance, and we knew now that our cultures don't have any resistance, and this indicating that there was no risk of transferring resistance to the pathogens. And that's all that I wanted to say.
Okay, time's up. So now is our Q&A session. Judges, any questions on this team?
It is a very interesting project, and which I have to say that it's very different from the rest of the projects that we are seeing, because you have incorporated some kind of a business element of feasibility analysis into the project, which is a nice thing. Okay. And I really have to ask, okay, whether you genuinely believe that this business is feasible. Have you ever considered any kind of risk that investor have to take when investing in kind of this sort of new kind of bioproteins production business?
Yes, I know about these risks because I made project management analysis, and this kind of work includes to find some risks for the investors, and I think that no, in my opinion, the main risks is that we won't have we won't have any farmers to buy our product. But I think that that according to places which have our product, it will be better than others, and that the farmers can buy. And and last but not least, that our product is more profitable, and it's not overpriced, and in this way farmers can buy it.
Okay, it's not exactly a risk, but anyway. And I'm also interested in the compatibility of your probiotics, which are also like microorganisms, with your kind of methanotrophic biomass, because for my understanding is that the methanotrophic biomass are how to say, it's anaerobic type of biomass, right?
Yes, and in this way we produced experimental fermentation, and we try to grow in aerobic and anaerobic condition, and in both way we got a high result, as I said, and also I can show again graphics. And there you can see the results in experimental sample and in the control sample, and as you can see, the result is great.
Okay. Yeah. Sorry, Jack.
Okay. So may I ask one question is that how do you come up with the list of microorganisms to test in your studies? So there are many different species. How do you like shortlist this microorganism as a candidate?
Okay. Yeah, if I'm not mistaken, you want to know how I get it or how I. Okay. These microorganisms we got from different animal surface. It's a fish and it's an egg, and we made so to firstly to get a clear culture because we want to patent this culture in future, and also we want to know which cultures is better to use in our production, in our fish production.
Okay.
Thank you, student. So you may now leave the meeting room. See you tomorrow at the award ceremony. Bye-bye.
Thank you.
Hello, Team IBBC 35. Welcome to the Global Youth Science and Technology Boat 2021. I'm Kimton, the MC of this judging panel. This team is from Turkey. The project is Cover with Green Seeds, Store Food Safely. First, we would like to thank you for attending the judging honor today and for your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project, and you can share your PowerPoints or other materials. And we will remind you after 5 minutes, and you have to stop your presentation when the time is up. And there will be a 4 minutes of Q&A session after your presentation. Do you have any questions?
No, thank you.
Okay, so you can share your PowerPoint now.
Okay.
You can start anytime when you are ready.
Okay, we can start. Hello, I'm Mirsa Kalkan.
I'm Irtizam.
We are 11th grade students at the Diyarbakır Open Science High School from Turkey. As you know, it has become more important to use biodegradable films instead of plastic packaging and films. which cause environmental and health problems. We aim to produce a natural and antibacterial biofilm in our project. By producing biodegradable films, quince extract, we aim to extend the shelf life of the foods and offer alternative to use of plastic, which major environmental problem today.
Quince is a sticky, untested substance, so we aim to make a biofilm with natural and antibacterial properties of quinces. In our biofilm, we use quinces, which are less and unusable. In previous studies, it has been said that quince is a source of phenolic acid, antioxidant, and flavonoid. Based on these features, we want to utilize this waste in our project.
In our study, we first extract the quince extract in the laboratory and use a biofilm production stage.
Before making our biofilm, we performed antibacterial analysis with the extract we obtained from quinces. In order to determine the antimicrobial effects, we applied the agar-weld fusion method with nutrient agar medium. After 48 hours of incubation, 37 centigrade degrees, we measured the zone diameter around 12. As you can see in the table, these results show us that the quince extract has high antimicrobial activity.
We make our biofilm after antimicrobial study. So how did we do that? For our biofilm production, we mixed agar, gelatin, distilled water, and glycerin in a magnetic stir. At certain temperatures, as you can see in the figure, then we divided the perfilm solution to separate beakers and added quince extract different concentration, respectively, and mixed for another 5 minutes. We poured the film solution we prepared in thin layers into petri dishes, previously stripped with UV light and left to dry under film hood room conditions.
As a result of our experiments, we obtained a high quality of biofilm from quince. We conducted trials on cheddar sample in order to determine the food preservative properties of our biofilm without any additives. We formed 3 groups of cheddar samples to be used in the study as uncoated 100% quince extract, 15% quince extract. We kept the samples at 4 centigrade degrees for 15 days and performed both visual and antimicrobial analysis every 3 days. According to data in visual analysis, with uncoated cheddar cheese samples started to dry on the 3rd day and started to deteriorate on the 6th day. The best results was obtained from Vita 100% quince extract. It shows that oxygen barrier is effective and has an edible film features.
In our antimicrobial analysis, we first took samples from each control and experimental groups on the 5th, 10th, and 15th days. And we performed a pre-enrichment phase in a peptide water for the growth of microorganisms. Then we divided the deletion series of these samples. By taking 100 microliters from each sample, and we applied separate plate method for the microbiological medium of the nutrient agar, and we grow up microbiological nutrient agar in a cell environment. For bacteria count, we kept in the dishes in a 37 centigrade degrees over 48 hours, and at the end of the period, we counted the bacterial colonies and graded in the medium and evaluated air density. As you can see on the table, when the colony count results of coated cheddar cheese sample added with the extract prepared are compared with, the best result was obtained film containing 100% quince extract.
Sum up, when we evaluate the results of our study, we can say that our film coating containing quince extract can be easily used in the food industry by using some improvements. With the edible films to be produced, it will be possible for both the industrial and the food sector to develop in this area. It will be an industrial benefit for future studies to include scopes to reduce production costs. In addition, we think that our project will contribute to packaging of food in a healthy way and deliver the consumers within the scope of COVID-19, which the world is struggling with. We think that our project will help packaging of food reach the consumers safely. Thanks for listening.
Okay, here comes to the Q&A session. Any questions from our judges?
I think it's a very interesting idea to make a biofilm. So may I ask, like, how do you exactly coat the cheese? So you dip the cheese into the quincy extract or how?
Could you please repeat that again?
Yeah. So how exactly you coat your cheese with the biofilm? You dip it into the quincy extract?
We made it by applying the dripping progress to the concentration with coating. So we make a dripping.
Yes.
Okay. So is it water resistant like plastic, your biofilm, water resistant like plastic, or it will dissolve in the water, in the hot water, for example?
I'm sorry, but your voice is not stable and your stop it again. Could you please repeat that?
Yes,
yes, sure. So I just want to ask whether your biofilm is water resistant or waterproof like plastic, or it will dissolve in water.
We can water. We choose solution. Yeah, not being plastic. Soluble. It's in the water.
Soluble in the water.
Yeah.
So when you store it in the fish, so the water will dissolve your biofilm then. When you transfer storage, the water will dissolve your biofilm. Would it?
What? Could you explain? You're saying dissolved or
in the air dissolve your biofilm.
Dissolve, right?
Yes. So if your biofilm is soluble in water, moisture in the air.
In the water, and you can wash it and destroy, not being and you can destroy and you can wash the damage and you can eat them, not being
so you add
you can not need the cup. You just wash.
You just need a wash in underwater.
Yeah.
Can be washed underwater, so it may not.
Yes, and
it probably won't dissolve in water. But my question to you is, now I've seen your experiment. Your biofilms actually made from
gelatin
and gelatin.
Gelatin. Yeah.
Right? But
yes,
the queen seed, what's the function of the queen seed? The function of the queen seed is not involved in making the film. It's just antibiotics or antimicrobials, right?
Yeah.
Yes.
But we can add some things and gelatin, agar, distilled water, and glycerin. Then we make this biofilm.
Okay, so the film actually
not to be shaped by yet and quince.
Okay, because also in your report you mentioned that when you put the queen seed in water, it will make a viscous kind of solution. So when I read your report, I thought that your edible biofilms actually make from the queen seed.
Yeah.
But it's not. It's from.
And yes, we can make edible films.
Queen seed has a gelatin and eco-friendly.
We use queen seed in the because of
we use queen seeds because of it is a gelatin sticky properties.
Okay, so probably the queen seeds also help to form a component of the edible film. But like in your control, you also have a control that doesn't have queen seed extract, but it still can form a film. So it's because I mean the title of your project is called Covered with Queen Seed, and it's in actual fact, it's the agar and the gelatins that form the sheet.
But I explained this. We extracted first quincetin and we added just some provers and like this, because you know that quincetin have a gelatin, and actually we use this not being too much gelatin agar, just being for a biofilm, and we combination quincetin gelatin and clotting. Yeah, not just yet.
Hey, thank you, team. I think it's time's up now.
Okay.
So you may
later. Okay, maybe. I mean, it's a very interesting project. Well done, and I just want to learn more.
If you want, you can say more questions. We have a time. It's okay for us.
Okay, well, maybe later. We communicate later.
Okay.
Okay.
Thank you so much.
Thank you so much. Goodbye.
Okay, thank you, team. You may leave the meeting room now and see you tomorrow in the award ceremony. Bye-bye.
See you. Thank you.
See you.
See you.
Team IBBC 23, Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. This team is from South Africa. The project is Effectiveness of Hand Sanitizers. First, we would like to thank you for attending the judging honor today and for your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your projects, and you can share your PowerPoint or other materials. And we will remind you the time after 5 minutes, and you have to stop the presentation when time is up. And then there will be a 4-minute Q&A session after your presentation. Do you have any questions?
No, I'm fine, thank you.
Okay, so you can share your video or your PowerPoint now. You can start anytime if you are ready.
Thank you. Hi, I'm John Ripalser from Hoërskool Waterkloof, and I would like to tell you about the effectiveness of hand sanitizers. So since the outbreak of the coronavirus, there have been different measures put in place to protect us from the virus and to stop it from spreading. One of these is to clean your hands regularly by soap and water or hand sanitizers. And I've always wondered how effective the hand sanitizers are and if you can make your own at home. So I created my hypothesis. Different types of alcohol kill microorganisms and can be used in hand sanitizers. I started my project with my research about the different types of alcohol and the bacteria. I then sent out a questionnaire to my friends and family. I then did my practical trial, but instead of using a virus, I used a bacteria, as testing on virus requires a controlled laboratory and can be hazardous. Then I got the opinion of an expert. So these are the two competitors, is the hand sanitizers and the soap and water. Of course, the soap and water removes all the microorganisms and cleans your hands, but they are not always available, while the hand sanitizers can be carried with you, but it doesn't remove all the dirt. And you also need 60 to 80% of alcohol to be effective. As you can see, there are 3 different types of alcohol, and the longer they get, the more effective they are. Then you have your bacteria. A bacteria can grow when it splits into 2 identical daughter cells, and the daughter cells are exact replicas of the mother cell. You also get your colonies, in which the form they grow, the elevation, and the margin. I got 26 respondents on my questionnaire that I sent out. On the first question that I asked them, I asked if they prefer soap and water or hand sanitizers. Most of the respondents prefer soap and water, while 95 of them actually use hand sanitizers as it is easy to carry with you. I also asked them if they knew the correct ingredients for a homemade hand sanitizer. Most of them knew, but not a lot of them knew the correct combination to make an effective hand sanitizer. I asked them what they think the aim of the hand sanitizer is. Most of them did think that the aim is to kill microorganisms, but some of them, like 6% of the people, thought that it is to replace soap and water. I also asked them how long they should wash their hands to kill all the microorganisms. Most of them said that you should wash it for 20 seconds to kill all of them, while some of them thought you should wash it longer than 30 seconds. I then did a practical trial to prove all my theories about the hand sanitizers. First, I made my homemade hand sanitizer using isopropanol, aloe vera gel, and essential oils. I then did the growth of bacteria. I cleaned my hands, and then I infected my hands, and I did a swap. I did culture to start the culture growth. I treated my hands with one of the 7 treatments. I swabbed my hands again, and then I started the culture growth. The treatments that I used would be soap and water, my own homemade hand sanitizers, and 5 other various store-bought hand sanitizers. I used agar Petri plates to do the culture growth. Of course, the soap and water worked the best, but the homemade hand sanitizer worked the best between the hand sanitizers. But between the commercial hand sanitizers, hand sanitizer 3 worked the best. I then went and made 3 different types of hand sanitizers with the 3 different types of alcohol. I then tested the effectiveness of them all. And methanol was the most effective, followed by isopropanol and ethanol. I expected ethanol to be more effective than isopropanol, but the quality of the isopropanol is unknown as it was store-bought and not from a laboratory. I then tested the evaporation rates of the different types of alcohol, and it came out methanol, ethanol, and propanol. Methanol has the shortest evaporation time, and it proved to kill the most bacteria. I then got the opinion of an expert, Henry Langerveld, biochemist, master student at the University of Pretoria. To sum up most of his answers, agar tests are good for testing bacterial growth, but not for the identification of the different types of bacteria. Agar tests are relatively cheap to conduct. More accurate tests are much more expensive. Hand sanitizers are effective if they contain the required 60 to 70% of alcohol. I have my different types of deductions, but the first one is the most interesting. Hand sanitizers kill microorganisms and can be used as a replacement for soap and water. Thus, my hypothesis is correct. Different types of alcohol kill microorganisms and can be used in hand sanitizers.
Okay, so here comes to the Q&A session. Any questions from our judges?
Okay.
So maybe I start first. And very nice presentation and also very interesting project, which is actually very close to life and close to our daily life. I just would like to ask, perhaps, can you show us more all the photos that you have about the bacterial culturing? Yes, I just want to, because I mean, it's not very clear how you did your bacterial culture. And I just would like to ask you whether you heard of the term aseptic technique.
No, I've never heard of that.
Okay, and that's to prevent contamination of your kind of culture plate when you open it. And I understand that from your reports that, I mean, you sterilize everything, okay, but at the moment you take off the lid of your culture dish and kind of dust from the ceiling everywhere may fall onto your culture medium. And if they contain bacteria, then you incubate it, they'll grow. And I just would like to ask whether you have done any prevention about those things.
We have tried to prevent them by when we made them, and then we would close them as quick as possible to prevent any extra bacteria from the air to get in. That is also why we have a controlled petri plate which doesn't have any bacterial growth in it, just to show that they aren't completely clean when you start. And we don't open them when we inspect them. As you can see, we just keep them closed to prevent any extra bacteria to get in. And usually the bacteria takes 3 to 7 days to grow, so you won't see them immediately when it touches the agar.
Okay, very good. Okay, and my next question is a bit like you measure the volatility of the 3 kind of alcohol. And of course, I mean, isopropanol is the least volatile. And have you done any experiments to see how long the kind of disinfection effect of the alcohol on your hand how long can it last?
I tested the rate of evaporation. Yeah.
Okay, so do we need to spray our hand like all the time?
If you that depends on what you touched. But if most of the time, if you have soap and water with you, it is better to use that than hand sanitizers. But if you don't have soap and water, it is best to use your hand sanitizer.
Yeah. Okay, thank you.
So I may have some comment about the alcohol you use. So methanol is actually very toxic, so it is not recommended to be used in the hand sanitizer. So this is not an idea. I think ethanol and propanol is okay. Propanol is also medically used for disinfectant for disinfecting pads or hand sanitizer. So these two are okay. Methanol is too toxic, so don't use it in your home sanitizer. Don't distribute to your friends. Okay.
Yes. When we did use the methanol, we used it on my hands, and then I immediately had to go wash my hands to get rid of the methanol to not make it toxic or to hurt myself.
Okay, thank you, student. So you may leave the meeting room now, and we will see you tomorrow at the award ceremony. Bye-bye.
Thank you.
Live now. Hello, Team IBBC 25. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of the judging panel. This team is from Italy. The project is a sweeter carnation, also more colorful. First, we would like to thank you for attending the judging honor today and for your efforts made in the past few months. Today, we are glad to have Professor Lam Hongwah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or other materials. We will remind you at the time after 5 minutes, and you will have to stop your presentation when the time is up. And after that, there will be a 4 minutes of Q&A session. Do you have any questions?
No.
If no, you can share your PowerPoint now. You can start anytime if you are ready.
Yes, we are ready. So, good afternoon. We are Antonio Gaspa
and Fabio Sanna.
We are from Italy and we are going to present our project. Is a sweeter carnation also more colorful? Our project has 2 objectives. The first is to evaluate the color intensity of purple carnation immersed in aqueous sucrose solutions. And the second is to confirm the visual observation by image processing. Our first objective reflects the chemical part of our project. We started from the study of the color flower that is due to anthocyanins, pigment that give colors from red to purple to flowers. We also studied the chemical structure of anthocyanins, in which is present a molecule of a sugar, in general glucose. And here we can see in the image the color of the flower we used in our experiment. So how it worked? In our experiment, we immersed some carnation in tap water and we used them as control or reference flowers, and the other in 100 milliliters of aqueous sucrose solutions at 3 different concentrations, 2, 6, and 10%. During the experiment, we took color photographs of the flowers. After 24 hours, we observed that the color intensity of the treated carnation is increased, and which is on the left. But we didn't observe this in the control one on the right.
So, for the second part of the project, we have the goal to confirm the visual observation by image processing. With the help of Dr. Sinis, we processed a matrix corresponding only to the red and blue channels in the RGB panel. This is due to the natural color of the carnation, which is purple, a sum between red and blue. Then, for each pixel, to extrapolate the colorimetric information. How we worked? First, we started by uploading the different photos inside a script, homemade script. Then, we removed the green channel contribution from the photographs. After this, we removed the background, which are pixels not representative of the flower's petals. After this, we constructed a diagram in memory, in which we uploaded the different data from the photos to then obtain a 2-dimensional representation of the diagram. Here, we have in the y-axis, the color intensity of the blue channel, and in the x-axis, the color intensity of the red channel. The bar at the side is representative of the normalized frequency of the pixels. After this, we've made the 3D representations of the diagrams before and after the treatment and of the control flower. This is the before the treatment. Here we can see a red zone, a dark zone, and a diagonal representative of the purple color of the carnations. After this, this is the treated flower after 24 hours. And here we have the purple zone and the dark zone that has almost vanished, and the red zone that has increased in frequency. After this, there is the 3D representation of the control coordination. Here we can see a similar situation between the control and the starter of the project. Okay, so this is how we analyze the images side by side. So the upload of the image, the removal of the green channel contribution, the removal of the background, the construction of the diagram in memory, the construction of the 2D representation of the diagram, and the 3D representation. And these are the 3D representation compared side by side. So, in conclusion,
we can say that yes, a sweeter carnation is also more colorful because sugars have an effect on the biosynthesis of anthocyanins when accumulation of this pigment in the basis of petals of treated flowers.
And by analyzing the 3D diagrams, we can confirm an increase in the red channel between after the start and after the treatment.
So this is our project, and thank you for the attention.
Thank you for attention.
Thank you, students. So now we are going to our Q&A session. Any questions from our judges?
Right now, no.
Maybe I can start my question first. So in your experiment, you use a smartphone to record the flower colors, right? But I don't know, cell phones, they always have some program that help you to automatically correct your colors, make some modifications or of a picture. So have you considered like how consistent your cell phone produce the picture? Are they consistently take the same picture? And also the light source and the environment affecting your analysis?
Yeah, that's actually a pretty big part in our project because we have also noticed this fact that cell phones generally tend to correct the images. In fact, they are not professional pictures. If we had the possibility to use a professional camera and some set up lighting in a more professional way, of course, our project would have turned more professional. Unfortunately, we didn't have this the camera.
Mm-hmm. And so also you speculate that the increase of color is due to the increase of anthocyanins. So how do you prove your hypothesis? Is there any experiment you can do? Or how do you explain your observation? So you provide an observation, but how do you explain your observation? With some hypothesis, with some mechanisms?
Sorry,
can you explain why the sugar water will like increase the color of the flower?
Yes, we know from scientific literature that sugar can improve the quantity of anthocyanins, which are pigments in some flowers. Mm-hmm. And these studies are about the influence of glucose. We used sucrose to purple carnation. We didn't demonstrate this in our project because we couldn't work in laboratories. We can only correlate the color changes of the treatment with an increased quantity of these pigments.
Okay.
So may I ask how many flowers did you test?
And we used 9 flowers.
So 9?
Yes, 9.
Okay, so because I mean, it would be nice if we are able to really see some kind of statistical comparison. And but for the time being, I mean, I read your report. I like it very much because I like the way that you use your mobile phone camera to do experiment, and you also have this and use MATLAB and to write like a nice image processing program routines to analyze data. That's something that we always like our students to be able to do. So good work. Okay, but I mean, it would be nice if you are able to really show a really a bouquet of flowers, okay, and to prove to people that it is not just kind of one or two flowers is happen all the time, happen every time. But I think my wife always told me that she adds sugar into the vase. I did not understand why. I understand now.
Hey, thank you, students. So you may leave this meeting room now, and we'll see you in our ceremony tomorrow. Bye bye.
Bye bye.
So we are now going to on live now. Hello, Team IBBC 29. Welcome to the Global Youth Science and Technology Boat 2021. I'm Kimton, the MC of this judging panel. This team is from Sweden. The project is the antimicrobial effect of beehive products. First, we would like to thank you for attending the judging online today and your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project, and you can share the PowerPoint or other materials. We will remind you the time after 5 minutes, and you have to stop the presentation when the time is up. And there will be 4 minutes Q&A session after your presentation. Do you have any questions?
No.
No, you can share your PowerPoint now. And you can start anytime if you're ready.
Yes. So my project is the antimicrobial effect of beehive products. Antibiotic resistance is one of the biggest public health challenges of our time. Infections caused by antibiotic-resistant bacteria are becoming increasingly difficult to treat, and in the worst case, completely impossible. Approximately 10 million people will die each year by 2050 if no action is taken. Antibiotic resistance threatens not only our health, but also the economy. A possible solution to the problem is to reduce the use of antibiotics, and this can be done by examining the antibacterial effect of the beehive products. If they prove to have a sufficiently high antibacterial effect, some antibiotics used today could be replaced by these in the future. The purpose of this study was to investigate whether the beehive products honey, propolis, and royal jelly inhibit the growth of gram-positive and gram-negative bacteria. Some delimitations were made on the work. The study was performed on gram-positive and gram-negative bacteria, but there was no knowledge about what species of bacteria it was. The study was also delimited by using only one type of honey, propolis, and royal jelly. Thus, the results can be misleading. The study was performed by first culturing the bacteria on agar plates. To be able to identify whether the grown bacteria were gram-positive or gram-negative, a gram staining method was then used. The identified bacteria were transferred to new agar plates where the examined products were also placed. After about 48 hours in the heating cabinet, the inhibition zones could be measured. The study was conducted twice. During experiment 1, the products were placed directly on the top of the agar. This affected the results because honey and propolis were diluted as they are liquid. In the next experiment, the agar well diffusion method was used instead, where the products honey and propolis were placed in the wells. The study shows that propolis has the greatest antibacterial effect, as it is the only product that formed clear inhibition zones. Propolis is also more effective against gram-positive bacteria, as the inhibition zone was 2 mm larger on gram-positive than gram-negative bacteria. Honey did not form any inhibition zones, but it can be observed that it inhibited the growth of all bacteria that came in direct contact with the honey. This result can be explained by the osmotic properties of honey. Due to its high sugar content, honey has a very low water potential, which results in the water diffusing out of bacterial cells, which in turn can lead to the bacteria dying from dehydration. The royal jelly gave different results on the 2 experiments that were performed. Like honey, the royal jelly inhibited the growth of bacteria that came in direct contact with the product in experiment 1. During experiment 2, gram-negative bacteria were not affected. There is a risk that 2 different bacterial species were used. The genetic material may have differed between the bacteria in the experiments, and thus the bacterium grown in experiment 2 may have been more resistant to royal jelly than the bacterium grown in experiment 1, even though both are gram-negative. Whether these products can be used instead of antibiotics is difficult to say. There are a number of sources of error that should be taken into account in the next attempt to improve the study. In future studies, I would like to use several different specific bacterial species to be able to compare the results. It would also be interesting to investigate how antibiotic-resistant bacteria are affected by the product. Thus, it can be found out if these products could be used against infections caused by bacteria that are resistant to antibiotics we use today. In addition, I would like to perform the study on several different varieties of honey, propolis, and royal jelly to be able to observe if some are more antibacterial than others. The products would then be taken from different geographical areas, since the chemical components of honey and propolis depend a lot on the plant kingdom in the area where the bees are located. And that's all for my presentation. Thank you.
Thank you. Here we come to the Q&A session. Any questions from our judges?
So what is your conclusion? Is the beehive products really able to replace antibiotics?
As I said, it's difficult to say right now. I need to do more studies on it. But the conclusion is that propolis could be used in slightly larger infections, as it nevertheless shows the high antibacterial effect. And honey, thanks to its osmotic properties, it could be used on like open wounds to prevent infections.
Yes, that's my next question. So your conclusion is that honey can be used to treat wounds?
For example, yes, to prevent infections, because it can kill the bacteria thanks to its osmotic properties by diffusing out the water of the bacteria.
Yeah, I remember somebody told me that before, that honey can be used to dress wounds, and your results seems to prove that.
Yeah.
And so can you name some of the nasty kind of pathogens that are gram negative?
Can you please repeat the question?
So your results show us that maybe even propolis and raw jelly it's not able to deal with gram negative bacteria compared to gram positive bacteria. Okay, so can you name any kind of nasty pathogens that are gram negative?
Gram negatives have outside the cell wall that wait a second.
It's okay. I just want to know any kind of a common pathogen that's known to infect, cause infections are gram negative.
Oh, I don't know right now. No, sorry.
It's okay.
Hello, Dr. Wang.
Okay, so may I ask, how do you define the inhibition zone? So how do you define the inhibition zone in your agapase? So in one sample, I noticed that it doesn't look like inhibiting the bacterial growth, but it looks like a contamination. The propolis.
Yes, it was an inhibition zone around the wall that I placed the propolis. I measured it with a roller, and it was 22 millimeters.
Mm.
It was no bacteria in that area.
Yeah, but in your propolis sample, it seems that there is not pure from bacteria, but there's also there actually something growth on the paste.
Yeah, it's a bacteria.
Can you go back to the slide?
Yes, of course. Is it this one you mean?
Yes, one. Yeah, it looks like there's something growth on the paste.
Wait a second. So this one is bacteria, and this in the center, this is propolis.
Mm-hmm. And what about the ring? Yeah, this ring. It seems like there's something.
No, that's nothing there.
It's nothing there?
May seem so in the picture, but there's nothing there.
Okay. Okay, thank you.
These brown things, this is propolis.
Okay, thank you.
Okay, thank you, student. So you may now leave the meeting room, and we will see you tomorrow at the award ceremony. Bye-bye.
Thank you. Bye.
Going on live. Hello, Team IBBC 21. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. This team is from Ireland. The topic is toxicological investigation of pesticides using paramecium as a model organism. First, we would like to thank you for attending the judging online today and for your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project, and we will remind you at the time after 5 minutes, and you will have to stop your presentation. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No.
If no, you can start sharing your screen or you can start your presentation.
Okay, I decided that I'm gonna talk about my project. So basically, my project is a toxicological investigation of pesticides, glyphosate, using paramecium as a model organism. My project investigates the effect of these pesticides on the survival and growth
of this single cell paramecium. Paramecium is a freshwater protist and is fundamental for the water environment, especially in small ponds or lagoon. My interest in developing about the topic developed during my study in geography and biology, and my growing awareness of the use of chemical in farming and gardening. As I live in Ireland, agriculture is a really important sector. Therefore, I decided to investigate more and raise awareness as well. And I found that the glyphosate herbicide is intensely used, as I said, and its effect can be measured on the wet environment. So I decided to observe the change in number of paramecia, their speed and size, and in 3 different time frames after 0, 24, and 48 hours. And at 5 different concentrations from 0.01 to 0.04 grams on the decimeter mass free squared. And basically these concentration are more than the Irish law, but at the same time less than the quantity found in rivers in Europe. So sometimes the quantity are even higher in a natural environment. I then use a microscope and per 10 object lens, a hemosiderometer and a camera to record the live paramecia, and subsequently I count them and measure their speed and their length. Through my investigation, I've said that the number of paramecia in 24 and 48 hours of concentration of glyphosate has dropped significantly. The speed of paramecia after 48 hours in the highest concentration decreased by 15.99% with a p-value of 0.013078, therefore significant as is less than 0.05. The paramecia number dropped until up to 31%, and it was much lower than expected. Then, as I did control, as I did, I use also a control without a pesticide to check if the decrease in these parameters was due to the pesticides or in general to changing of the environment, because they were in flask. So I use a control as well. At the end of the analysis of my result, I found that glyphosate, therefore, is extremely toxic for the paramecium and for this essential microorganism, and his role in the water ecosystem, because as I said before, the paramecia are fundamental for water ecosystem, as they play as food, but at the same time as cleaning, they clean the environment as well. And as a student, I was only able to observe the effect on paramecium. While a future study would be really interesting to actually measure the effect on the entire ecosystem, because if only on one single cell can actually do this herbicide can have this effect, studying on the entire ecosystem could reveal much more. And I found particularly interesting my investigation as the herbicide are fundamental for our society, and as we are 7 million people in the world to produce is not food for everyone, but at the same time is necessary to admit that
okay, time's up, student. So here is the Q&A session. Any questions from our judges?
So very interesting study. So may I ask, how do you determine the concentration of glyphosate used in your experiment? will it be too high when compared with the new situation?
How I decided the concentration.
Yes.
Okay. Basically, what I did was to look online on European registers. How the average the quantity found in rivers near place of agriculture, and at the same time to study the law of the maximum permitted. So I did the average between the law of the European members and the quantity that are founded in rivers. So therefore, the effect obviously. Obviously, the quantity of permitted by the law is not toxic, but I measure that the quantity founded in river can be toxic.
So in the test, you're using the paramecia to test the toxicity. In the short term, it's kind of acute toxicity. So have you considered would that glyphosate could lead to long-term effect beyond the 48 hours?
No, sorry, can you repeat the question? You asked me if after 48 hours, yeah?
So beyond, in addition to acute toxicity, so do you think the glyphosate will have long-term effect to the paramecia beyond the 48 hours?
Oh, absolutely, absolutely. So we measure that their growth, the quantity in their colony decrease, and the paramecia that survive, there will be much weakness, more weak, and at the same time they will be not able to reproduce, and one thing really interesting about the paramecia is that their life cycle is really short. Therefore, I was able to see what we can consider the long effect on only 48 hours. But probably if I measure for a longer time, definitely we would be able to prove that are much toxic than in 48 hours.
Okay.
So very nice work. Congratulations. And I like your project a lot because you use a kind of free-moving microorganism for your toxicity.
Yeah.
And you are able to add one dimension of measurement on top of just survival. Okay. And I can see that you measure speed, movement speed of paramecium as a parameters for your toxicity test. I just would like to ask whether you also consider using some like tracing software.
Oh yeah, absolutely. Yeah.
And have you already done that?
Okay, basically, what happened? I try using 3 different software on my computer, but as the computer wasn't able to identify the paramecia, but identified every single particle moving, for me was a problem then to understand what was what. Therefore, I found by myself a method by hands, and and that's how I did. So the computer didn't work. Meanwhile, my method that I invented worked. So yeah, I tried.
Are you able to just sting your paramecium so that the software can trace them better?
Oh yeah, the only problem is that the quality of the camera was good enough for me to understand what was what, but for the computer I tried many times and it just couldn't. Yeah.
Okay. But there are we are using like animal behavior tracing software, but of course we are looking at zebrafish. They are easier to spot.
Yeah, they're probably even bigger. It's like single cells, so they're really small.
Yes, if you are able to trace the movement of paramecium, how they move or they like move in erratical path, your toxicity study will be much more interesting.
Oh yeah, thank you. I will do that in future because I would like to continue my research.
Okay, thank you, student. I think that
Thank you very much.
So you can leave the meeting room now, and we'll see you tomorrow at the award ceremony. Bye bye.
Thank you. Bye.
I'm now going to on live. Hello, Team IBBC 05. Welcome to the Global Youth Science and Technology Bowl 2021. I'm Kimton, the MC of this judging panel. And this team is from Hong Kong. The project is Synthesis of Nanoparticles for Nitrite and Nitrate Decomposition in Wastewater. First, we would like to thank you for attending the judging online and for your efforts made in the past few months. Today, we are glad to have Professor Lam Hong Wah from City University of Hong Kong and Dr. Jack Wong from the University of Hong Kong to be the judges of the competition. Please note that my window will turn into a countdown timer when the judging officially starts. You will have 5 minutes to present your project with screen sharing of PowerPoint or any other materials. We will remind you the time after 5 minutes, and you have to stop the presentation when time's up. And there will be 4 minutes for the Q&A session after your presentation. Do you have any questions?
No, we're clear.
Okay, if not, then you can share your PowerPoint now. And if you are ready, you can start your presentation.
Good afternoon. We are pleased to present to you our project, synthesis of nanoparticles for nitrite and nitrate decomposition in wastewater. The improper treatment and discharge of wastewater with nitrogenous compounds causes severe environmental harm. We therefore aim to use recyclable materials to synthesize nanoparticles and determine the most effective method to decompose nitrites and nitrates and reduce the harmful effects of water pollution. Our project is divided into 4 parts. In part 1, we synthesized nanoiron. In part 2, we synthesized nanosilver. In part 3, we used our nanoparticles and used hand warmer powder to decompose nitrites and nitrates. In part 4, we prepared a nitrite filtration device.
Starting with part 1, we used reducing agents in banana, lemon, mango, and orange peel, black and green tea, and sodium borohydride to synthesize nanoiron. The high concentrations of polyphenols in fruit peels and catechins in tea act as reducing agents and capping agents to reduce iron 2 and iron 3 ions into nanoiron. Sodium borohydride also acts as a reducing agent. Fruit peels and tea bags were heated in water at 80 degrees Celsius to give fruit peel extracts and tea infusions. The extracts, infusions, and sodium borohydride were added to iron 2 and iron 3 salt solutions respectively. Nanoiron was then filtered and dried. Tea gave a higher yield than fruit peel, and black tea with iron 3 gave the highest yield among the 14 combinations. Dynamic light scattering showed that synthesis was largely successful, with most nanoiron having a size of 260 to 720 nanometers. Similarly, in part 2, we added black tea, green tea, and sodium borohydride to silver nitrate solution as a source of silver ions to synthesize nanosilver. Sodium borohydride gave the highest yield, and dynamic light scattering showed that synthesis was successful.
In part 3, we decomposed dissolved nitrites and nitrates with our nanoparticles and used hand warmer powder. They were added to nitrite and nitrate solutions, stirred for 1 hour, and then filtered. Nitrite and nitrate concentrations were then tested by commercial test kits and measured by colorimetry. In nitrite decomposition, nanoiron synthesized from iron 2 with green tea, which had a size of 700 nanometers, was the most effective. And adding used hand warmer powder enhanced this effect. Nanosilver was ineffective. In nitrate decomposition, nanoiron synthesized from iron 3 with black tea, which had a size of 400 nanometers, had the best effect. And adding used hand warmer powder reduced this effect, while the powder alone was effective. Nanosilver was ineffective. We propose that the synergistic effect brought by the used hand warmer powder is due to the iron 2,3 oxide present, which serves as a corrosion promoter, facilitating electron transport from zero valence nanoiron to nitrites, hence decomposing nitrites more effectively. In terms of production cost, the nanoiron synthesized from iron 2 with green tea cost $0.3 per gram, while the nanoiron synthesized from iron 3 with black tea cost $0.1 per gram.
In part 4, we made a filtration device with an inverted plastic bottle filled with the most effective nanoiron and used hand warmer powder. We ran nitrite solutions through the device and tested the filtrate and control with the nitrite test kit. The drop in color intensity indicated that our device is an eco-friendly way of removing nitrites in water. We believe that green synthesized nanoiron is largely effective in degrading nitrites and nitrates to lessen water pollution, with the added environmental benefit of reusing waste materials. Moreover, we found that used hand warmer powder plays a substantial role in decomposing nitrites and nitrates. Therefore, upcycling the powder can be a method of improving the environmental friendliness and cost effectiveness of green water treatment. This is the end of our presentation. Thank you.
Thank you, students. Now we are going to our Q&A session. Any questions from our judges?
So may I ask a question first? So thank you for your presentation. It's a very interesting project. So I have two questions for you. So in the experiment, you show that different starting material produce iron particle with different color. So what this color implies? If all these are iron nanoparticle, why do we have different color? Can you hear me? Can you hear me? Sorry.
Sorry. Yeah, I think we heard you. Regarding the color of the nanoparticles, we actually didn't do much further investigation into how the color would affect different, like the nitrite and nitrate decomposition ability of the different nanoparticles. But we believe, like in contrary to the colors, we believe that the size of the nanoparticles and the capping agents used were played a bigger role in determining their nitrite and nitrate decomposition ability of the nanoparticles instead.
Yes. Also, my second question is that it seems like in the nitrate and nitrite decomposition test, seems different iron nanoparticles give different activities. So why this will happen if these are all iron nanoparticles? Is that due to the impurity or other materials in the extract?
Sorry, we don't really understand your question. Could you?
Oh no. So in your test, you test the decomposition ability of different nanoparticles obtained from different reactions, right?
Yeah.
And they show different activities. But my question is that if all these products are iron nanoparticles, they should have the same activities, but it seems not. So what is the reason behind?
We propose that there is a relationship between the size of nanoparticles and their effectiveness in decomposing nitrites and nitrates. So from our experimental results, we found that iron nanoparticles made from green tea have a larger size and perform better in nitrite decomposition, while those made from black tea have a smaller size and perform better in nitrate decomposition. So we believe that it is because of the fact that green tea and black tea contains different kinds of reducing agents and capping agents. The nanoparticles will have different organic coatings and have different sizes. So we believe that nanoparticles with different sizes will have different reduction potentials. So when we use them to treat different oxidizing agents like nitrites and nitrates, the nanoparticles will give different degradation effects.
Okay, thank you very much.
Very nice project and nice presentation. May I ask, well, you attribute the decrease of nitrate and nitrite from your test solution to degradation, to decomposition. And from your presentation, and you implied that the nitrite and nitrates got reduced, right? And have you really proved that? How can you rule out adsorption where the iron particles, nanoparticles, kind of, or the nitrate and nitrite just adheres onto the surface of your nanoparticles so that they disappear from the solution?
We can measure the pH to prove the redox chemistry. If the fall in nitrite and nitrate concentrations were due to degradation effect, pH of the resultant solutions would increase. If not, the pH would remain unchanged. Also, we can use mass spectrometry to prove that nitrite and nitrate ions are degraded. If the highest mass-to-charge ratio is smaller, it shows that the ions have degraded with a smaller formula mass.
Okay, so did you monitor a change in pH?
We didn't do that in our investigation, but this could be a probable way of moving forward in terms of how to improve our project for further research.
Okay, yes. And I'm also curious why silver doesn't work.
The reason why our nanoparticles can degrade nitrites and nitrates is because of the reduction process. And we found that iron has a low reducing power. So the use of silver nanoparticles is not effective in reducing nitrites and nitrates.
Sorry, I believe my teammate meant to say silver has a low reducing power instead of iron. Yeah.
Yes. Okay. So you can check the reduction potential of silver plus to silver and iron to iron. Okay. Thank you very much.
Thank you, team. So you may now leave the meeting room, and we'll see you at the award ceremony tomorrow. Bye-bye.
Thank you very much. Bye.